Okay, my friends, as always, shocker du jour today about viruses, what they are, what they do to the human body, how specific are they? What do they know about a virus? Is it alive? Is it dead? They don't even know. I'm not kidding you. There's a lot they're just starting to learn. And they have, well, let's just jump into it. Let's just jump in. Okay, this is a brain teaser. We've all talked about viruses. Everybody said, oh, a virus, oh, that's just a little flu virus, a bug. Well, they're not bugs. Viruses, they don't know whether they're alive or dead. They claim they're like in the margin in between. And they take over the cellular machinery of other just normal cells, and they direct them to do their bidding. Now, where did they come from? How did they reprogram that cell? Where did they come from? They say they're in a capsid, which is like a little capsule. Well, where'd that come from? Now they're starting to understand. Well, this just came out a couple days ago. This is a new approach, uncovers cryptic endogenous retrovirus subfamilies in the primate lineage. Now, when they talk about primate lineage, first of all, viruses are very, well, not they're extremely specific, but they are pretty specific to certain types of creatures, certain species. Like we are, I believe, the only ones, hold on, I think we're the only ones that can get infected by measles and something else. Hold on. All right, here it is specifically right here. It says virus is often quite specific about the species and even the cell types they can infect. How do they infect them, first of all? But secondly, they're specific to the cell type. And, you know, quite often, right down to the cell types. But here's one example. Human papillomavirus, HPV, and the measles virus only infect humans. There's a reason for that. There's a reason for all these specificities. What exactly is that reason? All right, this is basically, you're trying to understand biology. They're saying these are the viruses, these little balls. And each cell has a membrane around it, and that's the body of the cell. The virus enters the cell. Well, that's fine. And then it opens up, and it becomes a virus. And it goes in and reprograms that cell, basically, to start creating its own viruses. So it takes over that cell and says, I want you to stop doing what you're supposed to be doing, and I want you to make new virus cells for me and go out and spread them around. That's the claim. Where did the virus come from? Well, we know they're coming out of cells once they're produced, but where did the original one that started the whole process come from? Well, let's just read this together. Viruses are generally considered non-living entities. They really don't know much about them. They exhibit some characteristics of life, such as replication, evolution. They lack essential features like cell structure, the ability to independently reproduce or metabolize. They're little inert chemistry sets. They're little inert, actually these are little inert programs. That program is identical to a ribosome package. However, it reprograms the DNA within the bacteria to be a new program to create new viruses. All right, that's what a virus does. But where does that original virus that has that program in it come from? We know that bacteria create ribosomes, but what creates viruses? Do bacteria create viruses? I'm not sure at this point, but the viruses are non-living, which is identical to what happens with these enzymes that are inside of ribosomes. But the ribosomes are created by bacteria. What creates the virus? Viruses are programs, but they're encapsulated just like the enzymes. They look to me like they're ribosomes. Let's dig into this. All right, basically they do not know where viruses come from. They say viruses originate from various sources. Well, and they evolve to infect other cells. Some theories, and these are just theories, these are guesses, suggest viruses arose from escaped cellular genetic material. Some DNA broke off, little bits and pieces went here and there. It just somehow got it loose. I don't go with that. Others propose they evolved from pre-cellular life forms. I don't see that either. And then they just go and say, oh, well, forget about it. Regardless of their origin, viruses have developed a mechanism to attach to other host cells. I like to find out what the origin is. That's where I'm going. So as they say, oh, forget about how they developed, but now we know what they do. They use specific proteins to bind to receptors on the cell surface. All right, so they bind on the surface of cells. And they enter the host cell. They dig through that membrane. They're digging through membranes. All cells, everything in your body is coated with a membrane. If these can get through that membrane, you get infected. Now, so it's just saying that they are, regardless of their origin, they developed a mechanism to attach to and enter host cells using specific proteins to bind to receptors. When I say specific proteins, people say, oh, well, what does that mean, protein? Well, I'll show you what it means. Anyway, they use the specific chemistry to bind to the receptors on the cell surface. Bang. Now, once inside, they hijack the cell's machinery to replicate and produce new viral particles. What they do is they come inside, and normally that cell or that bacteria or whatever it is going to produce, well, you know, what types of cells do they infect? Do they just infect any cell in your body? I don't think so. I think they infect certain cells. And once they hijack that specific type of cell's machinery, they replicate and produce new viral cells. But I believe probably only in that specific type of cell. You got all kinds of different cells in your body. I don't think they all get infected at one time by this particular virus. They're very specific to what they do attack. Now, this is I got to look into see if viruses attack all cells equally. I don't think they do. I think they attack certain cells, and they're able to reprogram those certain cells, but not every cell. Okay, I told you this is a thinking exercise. We are going to do this together. We're going to drag you through the mud with me. Now, I understand viruses, bacteria, cellular biology, genomes, the genetic structure, what turns things on and off, methylation. I understand a lot of this pretty deeply. However, viruses have always been an unknown. Where did they come from? Nobody knows, and they still don't know. So don't think I know for certain. I am going to work this through with you. All right, let's start with some ground rules. Every species of creature, it has a different set of DNA codes. That's not an unknown. That is fully understood. Now, some of them are mammals, some of them are birds, reptiles, anything, whatever. But you're going to have a set of DNA that has the program codes that say make fingers makes toes, make the whatever it is. That's the coded DNA, which is structural. It's like an instruction manual to build the creature and to maintain it, really. It's that amazing. It really is. And that's only a few percentage. of that DNA strand. These DNA strands are absolutely stunningly capable. Just a little clip of it can make everything happen for life. Just think about that. And if you knew the biology the way I know the biology inside, how deep it goes, it's just stunning. We're going to be down in the ribosome area, which is way down inside the cells. Every cell in your body has a membrane around it. Every single one, 100%, even your red blood cells, have membranes surrounding them. This is the most critical thing of all, is these membranes. All right, so we're going to talk about viruses, about membranes, about infections. What gets infected? There's certain types of infections that only infect your respiratory system. Other ones only infect your liver. Other ones only infect something else. So they can be pretty specific. That's got to be indicative of something. So we take that as a clue. All right, we take the fact that we don't know where they come from. That's a clue. But they claim they're packaged in a little ball, what's called a capsid. It's just a little, like little cellophane coating, and they open up into viruses inside of, I believe it must be the bacteria. And they take over that bacteria's program and split it out. Now, are they opening up inside of a regular human cell, and then the human cell actually creates it? I don't think so. I think the human cell, inside that human cell, the bacteria that's in there, is the thing that's creating it. We are loaded with bacteria. Loaded. All right, this is from the University of Queensland, Australia. And they're saying, what's the difference between bacteria and viruses? They're both invisible to the naked eye and cause your sniff, fever, or cough. So how can we tell the difference? And then it goes on about bacteria and virus all around us. They're everywhere. Can they invade you? Maybe. If there's 5 of you in one room and one guy gets invaded and the other 4 don't, why? Why shouldn't everybody get invaded if you're exposed to the same toxic stuff, the virus, the bacteria, whatever it is that's causing a disease or an illness or your sniffles? One guy gets it and nobody else gets it. Well, there's normally a reason for that. I find that there's a reason for virtually everything. So let's get down to what are viruses. I think we already talked about this, but this is what they have to say. Viruses are assembly of different types of molecules consist of genetic material. They're just talking about all it is strands of DNA and RNA. So they're saying it's either single or double-stranded DNA or RNA with a protein coat, which is the capsid they call it, and sometimes a layer of fat as the envelope. Now, they can assume different shapes and sizes, spacecraft designs, spirals, cylinders, ball shapes, spiky, little blobby looking things. Viruses that are enveloped with a layer of fat, now which is SARS-CoV-2, which causes COVID-19, can be more readily killed by simple hand washing. They say because it has a layer of fat on it. The soap disrupts this fatty layer and kills the virus. That's what they're saying. Now, viruses cannot reproduce. They're there, and once they infect you, that host reproduces. The virus itself can't reproduce. It says viruses cannot reproduce on their own. That's different than bacteria. Bacteria grow and thrive and just bubble everywhere when they're in the right conditions. When they're not in the right conditions, they will not thrive, and they will not, just be healthy and thriving and putting out all kinds of enzymes. So you will be immunocompromised if your bacteria is not in a good, comfortable environment in your body. So viruses can't reproduce. The viruses, they just can't reproduce. Bacteria can. So the viruses aren't considered living, but they can survive on surfaces for a varying level of time. Now, I have found that enzymes can last almost forever, because we have catalase, which is an enzyme, and all living things have catalase. That is a destroyer of extracellular oxygen. It just turns it into water. So hydrogen peroxide, H2O2, is attacked by catalase, which is an enzyme, and it pops off one of the oxygens. So what do you end up with? H2O. H2O. That's water. Your body loves water, and the oxygen bubbles away. That's why you have to have catalase, and that lasts just about forever. So at this point, what do we know about this? I will accept the fact that viruses infect good cells. Now, what good cells do they infect? Do they infect our human good cells, or do they infect bacteria that lives in these areas, like your lungs? And then your lungs get all infected, or your liver, and your liver gets some kind of infection, or some other area in your body gets infected because they're moving into an environment that they want to live in, because that chemistry there is good. Now, is it the chemistry of the cells that are living there, that are your cells, or is it the chemistry of the bacteria that's living there? And they take over the bacteria starts squirting out these viruses, which is what all of a sudden you get sicker. If you can't fight back, they go crazy. But you should have that bacteria that's also going crazy against it. Bacteria should just, because you got so much surface area in your body that has bacteria in it, you should never be overwhelmed by any of this stuff. If you were 100% healthy, this stuff would never get into you. It would never be able to get past your immune system. I don't think. There are some extremely, voracious, like attacking viruses, yes. But I don't think every single person that gets it. If you're exposed to it, I don't think 100% get anything. So something in certain people gives them the ability to withstand the attack by virtually any kind of virus. But not everybody has every kind of immunity. I see that as a deficiency somewhere in the enzymes you have to fight back. And those enzymes are created by bacteria, so I see a deficiency in a bacteria. And they have to live in your body, those bacteria, and they have to be in a comfortable environment. They have to have food that supports them. They have to be able to have niches that haven't been invaded and hit by antibiotics and toxins and all that stuff. Virtually everything in you tries to kill the bacteria that's in you. When you started taking these toxins and alcohol and smoking this and that, and intravenous drugs, and just about anything, antibiotics, different toxins in the soil, different herbicides, all that is trying to kill your bacteria, making it less able to fight back against these invaders. That's my position at this point. All right, this is about the relationship between bacteria and viruses and their infections, and they say they're often related. Well, we'll talk about that. Bacteria and viral infections are different, but they're often related, and they are treated differently too. Now, severe cases of viral pneumonia, which is from a virus, often end up with an associated bacteria infecting that site. This is particularly true with COVID-19. Up to 50% of the severely ill hospitalized patients have developed a bacterial infection. So despite COVID-19 being caused by a virus, antibiotics are really important to treat the associated bacterial infections. Once they start treating them with the antibiotics, they really should be replacing the probiotics in the gut, and I don't think that they're taking that into account as much as they should. And they may, I don't know, but I don't think so. It says, as antibiotic-resistant bacteria are an increasing global problem, researchers at IMB are investigating the surface activity of bacteria at the molecular level and have discovered how they elude the human immune system. The human immune system is powered by bacterial enzymes. If you don't have the bacteria, you will not have the enzyme, and they just can flow freely and do anything they want. Now, you may not have enough of the enzymes to overcome the power of viruses reproducing. They reproduce at a phenomenal rate, but so should your good bacteria create ribosomes that increase at a very rapid rate too. And you have much more surface area of your own good biology. So if you have those good microbes living in the right places, producing the right enzymes, these are never going to bother you. That's why there's 5 people in a room, 1 or 2 or 3 get sick, even with the worst illnesses, and the rest don't. They have something in their body that says, no, you can't come through here. And the only thing that does that is chemistry. And the only thing that works fast enough to do that chemistry is an enzyme, and the only thing that creates the enzyme is a bacteria. I go to the bottom of the ladder and I say, how do you start up that ladder? Well, you got to have some bacteria down here and some enzymes, or you can go up the ladder all you want. And that's what happens with the viruses. We need to get down here and say, okay, create some stuff there that they try to come in and kills them. That's all. And that is going to be a bacterial enzyme. We need a database to show what bacteria and enzymes you have in your body in a normal human being that is not having any symptoms of anything. And what is normal? And you're going to find out that a lot of people, they say, I'm healthy as and you know, I got to be honest with you, I've been healthy as can be. I take probiotics and Metamucil and a couple of little things here and there, nothing much. But the probiotics, that's the key. For me, that's my opinion. Anyway, and they're finding this replacing the bacteria in your gut is basically the solution to virtually everything. That is where all the digestive action happens in your body, is in your digestive system. It's as simple as that. You don't take in the right chemistry, you're in trouble. How do you get the right chemistry in you? You have the right bacteria to break down the components you eat. You eat a carrot, it doesn't turn into a muscle. Right? It doesn't turn into an eyeball. But, you know, they say you need some of this, you need some of that, and some of that. Well, yeah, well, how do you get it out of that carrot? You need enzymes to come in and go, and it breaks it down. And it does it just like that. They do more chemistry in 1 second than could happen in 1,000,000 years in a lot of cases. Enzymes are just, they are the cat's meow, and they are everywhere if you have the right bacteria. Don't have the right bacteria, they're not everywhere. You could have some of that right bacteria, not enough, you can be overwhelmed. You can have none of that bacteria, you pop like a cork. All right, so really in my mind, a bacteria creating a ribosome is virtually a virus. And when these viruses take over bacteria, they start squirting out ribosomes, which are the viruses. They do the same thing a bacteria does, squirting out ribosomes, which are good for you in most cases, and they start squirting out ones that are bad for you. It's just a reprogram. The virus goes in and says, hey, stop doing what you're doing. I'm reprogramming, shut the system down. Boom. Okay, start shooting those puppies back out again. This time they come out with this guy's DNA in it. And they go attacking all the other cells in you. So they try to take over that mechanism so they can, you know, set up shop there. They're invaders. All right, just going to go over this very briefly. It's very simple. Bacteria are living cells. They're living single-cell creatures, have the capabilities of consuming waste of all kinds of different types. They can reproduce by themselves. They actually produce enzymes, and in great abundance. Better said, bacteria are the factories that produce enzymes. All right, now, so let's go from there into bacteria enzymes. The enzymes are the chemicals. They're in little packages called ribosomes. All right, this is important to understand. Enzymes, they are not alive. They're just like viruses. They're not alive. But they have powerful, very powerful chemistry. Now, they're complex chemical packages called ribosomes produced by bacteria. They cannot reproduce. They don't eat, they don't poop, they don't reproduce, they don't do anything. They just float around, and when they're needed, they pop open. So they cannot reproduce or actually consume waste. They don't do that job. They speed up chemical reactions without getting used themselves. And like I said, in 1 second they can do more chemistry than what could be done in 1,000,000 years. All that chemistry would happen, but it would take 1,000,000 years. These enzymes are unbelievable. Now, don't forget this now. Enzymes are all proteins. Well, they're all proteins. I'll go along with that. And some enzymes attack proteins, all right? Because they break things. Therefore, enzymes' usefulness is limited by the digestion from other enzymes. So if you have some enzymes in you, basically I'm looking at those as the viruses almost, and they attack proteins and other enzymes and bacteria. All right, so you have to digest other things to create enzymes to attack those attacking enzymes. That's what it looks like to me. Now, I bring this up because it's not well understood. This layer right here is the fluid-filled highway that they never knew was here. This is from 2018. Meet your interstitium, a newfound organ. It's this layer. It coats everything in your entire body. Every different type of biology is separated by a membrane, and that membrane says you stay down in there because your pH is 7.5 and up here is supposed to be 3.2, and you guys mix together, you're going to all fester up and get infected and so forth. So if you need chemistry in here, let's just put it this way, that fluid-filled highway is your emergency system and your immune system. It sends attackers to attack invaders. It sends other things out through to deliver goods using transition metals primarily, and they carry little things around. They also do it in your blood, but this also is a very, very serious delivery system, and they actually show the stuff shooting out into the system being produced in this darker layer down here. Now, a lot of doctors do not know about this. Like I say, just 2018. This is not long ago. For medicine, they move very slowly. And this layer here squirts out whatever it needs, and it shoots down through your fluid-filled highway to go to attack invaders. And that's why you have all pus and everything coming when you have a real bad infection. And something's trying to kill your blood cells that are in here, and you got other things fighting back trying to kill those invaders. But infection, inflammation, you know what an infection is. That's because you're trying to fight back, and you see it inflamed and everything. That is a reaction. In most cases, it's short-lived, and that's good. If it's not short-lived, not good. That means your guys are not being able to come and do their job. All right, so let's take a look at what this looks like if you were right down looking at the blood cells and all the little blood cells traveling through and feeding the muscle tissue. All right, these blue ones are your veins bringing the blood back to be reprocessed. And this is your muscle tissue. Now, this, I believe, are neurons sending little nerve signals to do whatever has to be done. Obviously, it's an artist's rendition, but basically, this is the way the tissue underneath the skin is. You have to deliver blood and minerals and all the things your body needs. There's 1 million different little cells in here. They're not showing them, but there's a bazillion little tiny cells. And that's what these are bringing, and they're dropping things off. And they just keep going until they've run out of all the stuff, and then it ends up going here and heading back to your heart to get pumped around the body, to get recharged, get some more oxygen, filter out some of the stuff in your liver, kidneys, throw in a few secretions here and there, go through a couple of lab experiments in your body. Your body checks this stuff all the time. I mean, it's just amazing what your body can do if it's working right. But to work right, it has to, absolutely 100% has to have the right enzymes, which means 100% has to have the right bacteria. That's what we need, is a bacterial database. What the bacteria exists in these creatures. Then we can make a determination of why some people get sick and other ones don't. Sooner or later, you're going to fall into a pattern of saying, hey, everybody that's getting that sickness has this similar sort of a situation with these particular type of chemistry, which is bacteria. Now, getting back to that, we don't have to know every species of bacteria that's in here. Couldn't care less about that. Every bacteria that is in your body and every little bit of chemistry has a different color. You see it? When you take a picture of that with complementary metal oxide silicate receptors, you can take an image of this whole thing and tell how much of each thing is here, what colors, in what spectrum, and then put it through AI, look at that picture and say, we got too much of the blue and not enough of the orange, and the red isn't the right tint, it's off by a couple of nanometers or whatever they are. Here's the deal. This is what you're looking at. All of these different colors coming back are frequencies. And we're just bouncing them, and it tells us every single detail. Now, how much of that frequency? If you got too much of that frequency, you're sick. If you don't have any of this frequency down here, you're sick. And there'll be a database, we can track all that stuff. And if it works with the spectroscopy, which I can't see any way it shouldn't, you just have to be able to take a picture of your poop, send it in, and within a couple of minutes, it'll tell you what your baseline is. And then you go from there. That could be the solution to health. I mean, seriously, it could be that simple. Know what kind of bacteria you have to add. You don't want to add every bacteria there is, and good and bad and this and that, who knows what. And that's what's happening right now with the way they're doing it now. We need to have that database, and we need to have it not only here and there, but you need regional, you need where these people live, what kind of water they're drinking. You really got to set up a database where you can really filter it down, because you may find little populations that are just absolutely horrifying, and there is some. And I've been working on one right now that they have a terrible, terrible autism rate. It's like 1 in 22 or something right now in Northern Ireland, and they're trying to whittle that down. And I have some people that have been doing fabulous work over there, and they have treated many families, and they have helped them. And we need to be able to know what bacteria is missing, get that database going. It's not a big thing to do. It's going to require some coordination, working with people that want to solve this problem. This is just a horrifying problem. And if it is what I'm saying it is, the solution is absolutely simple. All right, here it is, hot off the press. Recent research suggests a significant link between a gut microbiome, which is the bacteria, and autism spectrum, with potential implications for both diagnosis and treatment. Studies have identified unique bacterial signatures, that means different strains of bacteria, in the poop of children with autistic diagnosis, hinting at the possibility of using stool sample testing for early autism detection. We need to supplement that gut bacteria and bring them back to health. That's what we need to do. Test them, yes. Find out what's missing, and then we can be able to supplement that and get the real living bacteria back in there so that it repopulates these missing regions that should be producing its own probiotics. Right now, we've been supplementing them, and it's been working. But your body should create its own probiotics through these good bacteria that's living in all of these specific places. Primarily the gut is where you're doing all this digesting and transferring all these particles here and there into the bloodstream and all that. However, that happens everywhere in your body. It's not just in your gut. It is primarily in the gut. But in all the areas where you secrete anything, the gallbladder, or, well, really everywhere, lymph nodes, all that stuff, they're working with bacteria and enzymes. They live in those areas. That's why you get cancer in a certain organ, because that particular organ it's lost its ability to fight off invaders. Let me show you what a cell membrane is. And it's going to make more sense when you think about, well, let's look at it. Okay, this is the Tour de Grande. This I showed you is a new organ, this specific layer right there. This dark layer under here is where the chemistry is done to spit out into this layer to go treat whatever it has to do around the body. And here it is when you're up close. There's that fluid-filled layer. Never knew that was there. This is skin or something above. It could be, it's fascia, let's put it that way. And below that, or skin or whatever it is the fluid-filled highway. And below that is where your membranes are they're all like this. Membranes are like this. And this is how I discovered this from working with mud fossils. There is a ton of layers. This right here is the fluid-filled highway. I believe that is it right there. And what happens is, you got a whole bunch of layers you're coming through up here. You don't see them as really distinct layers, but trust me, they are layers. And then when you get to here, you've just broken through this little spot here, and which is this layer, let's say. And now you're into the fluid-filled highway right there. And that is it. Now, below that is a whole series of places that create, this to squirt out there. Now, I don't know whether this was taken specifically to look at the response to an invasion, but it could very well have been, because normally I would think these are just sitting around, but they may be spitting them out all day long. I don't know, but these are spitting out. I believe these are ribosomes, bacteria, whatever they are, macrophages, and they are the ones that go through the body in basically the lymph. This is almost completely the lymphatic system. Very, very thin layer of very fine fluids. It's like this. And it surrounds everything in the body. Every single organ has, and every cell has its own membrane that surrounds it with the fluid-filled highway. So that means every one of them has bacteria down here, and basically, to my way of thinking, if it's a different type of cell or a different type of organ, it's going to have a different type of bacteria living under here, supplying protection through this membrane, because that membrane surrounds that organ. That's it. It just surrounds that one organ. And that could have a totally different pH than the thing it's bumping right up against to, the liver or whatever it is. So I'm saying that's why we have so many cancers. They're invading that layer. Now, when I say invasion, what does that mean? Invasion means you're supposed to have tight junctions. Now, I'm not sure where the junctions are in this. They may be down in here. I don't know. There's tight junctions in every cell. They're supposed to be held together and be able to flop around like that and do this and that. But you go like that, you're down into the good tissue. You're not supposed to be there. You see this? These are cellular junctions. That's the basement layer. And then you have this whole block sits on top of the basement layer, which is all of these junctions. You see this? This is a junction right here. Junction here. Tight junctions up here. Don't come past here. Stay out. Stay out. It makes this thing float around like a gooey substance, yes. But these different junctions say, don't come through here. Stay out. There are places that allow things through. But there's all kinds of little organelle-ish looking things, gap junctions and hemidosmiones. These are the new things that they just started talking about. Now, I've known about these for a very long time, and now they're talking about they just discovered these. I don't know why they just discovered them. This picture goes back quite a long time ago. The different types of cell junctions. Red dots, actin filaments, neighboring cells together, desmosomes. And where's the other ones here? Well, anyway, these here have that little bump. Well, you know what? Let me show you that thing. But I believe this is what they're talking about. Hemi, which is red blood, desmosomes. All right, and they're going down right into the red blood, I believe, right below into the basal lamina, and where there's going to be some bloody places to work with. But this is not new. They just, I don't know why they say it's new. All right, this I've been looking into this. For some reason, they claim they just found this. This in 2025. They're saying they just discovered it.
In 2025, scientists uncovered a previously unknown structure hiding inside human cells, an organelle called the hemifusum, discovered using cryogenic electron tomography, which freezes tissue and reveals three-dimensional structures with electron beams. The hemifusum sits between two vesicles and acts like a microscopic recycling center. It sorts, reroutes, and processes cellular materials, playing a critical role in maintaining cell balance. This is like discovering a new recycling center inside the cell, says Seham Ibrahim of the University of Virginia. If the hemifusum system fails, it could trigger cellular dysfunction linked to widespread disease. Researchers also observed that these structures behave unlike anything previously seen in known transport pathways, especially when tested with gold nanoparticles, suggesting they operate by new biological rules. While their full role remains under investigation, this discovery could change how we understand and treat genetic and degenerative diseases. The findings are now published in Nature Communications, marking the start of a new chapter in cell biology.
New chapter in cell biology. Hemifusum. I don't know why this is brand new. I thought they knew about this. Well, that's actually my mistake, although it's pretty confusing. These are desmosomes. All right? These are hemidesmosomes. The other ones they call hemifusomes. It's like these two are together or something. Maybe they are. I don't know. There's all kinds of things going on in here that nobody's ever paid that much attention to. But I'm going to tell you right now, as far as I'm concerned, whatever the chemistry is in your body from the enzyme, from the bacteria that keeps these cell junctions tight is your first line of defense. Once it gets through there and it's into your good layers down here, then, you know, you're at risk. You have to have the guys be able to come back here and attack that. Now, you are going to get attacked through your junctions here all the time. You get pokes and punctures and cuts and, you know, all kinds of, you know, everybody gets injuries, and they can be pretty deep. So once you're cut in here, your guys are supposed to send your hemifusomes or whatever they are to kill off all that stuff, you know, your enzymes, your proteins, all those things come over there in your lymphatic system and they attack. If you don't have them, there's no attack. And then that thing starts to grow down there. That's what cancer is. It breaks through here and it just starts growing. Nobody's here to attack it. Nobody's here to say, get out of here. And it's just, and there's 200 or more types of cancers, and every one of them attacks a different type of cellular membrane. Every membrane, in my mind, is going to be a smidge different. And I think I have found a little tiny organelle that's in the connection between each organ and where the fascia fits together. There's two little dots. And I have found them, once again, in my mud fossils. They're right there. There's two little dots right there. Hold on. You might even be able to see it while I even turn on a microscope. Probably not. They are there. Let me see if I can make this work. Hold on. They're right down at the very tip. If you got good eyes, maybe you can see them. There's 2 dots right there. And that flap, there they are. That's better. That's a good shot right there. See the 2 dots? That flap attaches to the next body part. And this little round spot here, that right there is the anchor. The anchor's this. And then the flap goes down, and they call that the venous flap. And it has veins. And you can see the black lines. If you can, I don't know if you can or not. There's black lines in here, and they bring the vein fluid down through, which is lymphatic fluid. It's not actually vein fluid. Anyway, that's, I can see this stuff in my microscope better than they can see it in the doctor's office. Because some of the stuff I have is just absolutely stunningly large, and it's just the way life is. I didn't make those rules. They came with life. I didn't do it. Now, I've done an awful lot of work on this. I've worked with a lot of people that are some of the only people that are working with fascia and understand fascia. And that is fascia. That's fascia right there. That's the coating that wraps whatever that was, a muscle or whatever. And this is the attachment between this bundle of muscle or organ or whatever it is up to wherever it attaches to and locks in up above. This had the same thing. This right here is basically the same thing as that. There's a tag right here, which is that latch. That little round latch latches in here, and that's the real strong spot. And then off comes the flap, and then the vein and artery go out through that flap. Other than that, it's an enclosed rubber bag with a complete layer, fluid-filled highway surrounding that particular organ. Like this, well, just take my word for it. It's like a rubber balloon that surrounds the organ, but it has to have some fascia coming in, fluid coming in, some going out. And that's where that little flap comes, the 2 little tags that I showed you before, coming right out of this, the bottom of this little latch right here. And you see the black? That's vein. Whenever you see that, that's vein. And that's where this tag is, right up here. This is a heavy-duty, gnarly thing. Tough, tough, tough. See how tough that spot is? I don't know. And then it comes down, wraps right around to there, and those 2 little dots are at the end there. Whether you can see them or not, je ne sais pas. But this is muscle. And that is the fascia. Just like I'm showing you here. Okay, this is like a refresher course if you've been into biology. Bacteria, right here, have their own DNA, and they also have a set of codes that says I produce a ribosome. And every species has its own ribosome, every type of bacteria, and there's 75,000 enzymes, and ribosomes are the package that is the enzyme. So we have these, and when they create that ribosome, that little ball I showed you, that little ball comes out, and a trigger triggers it, whatever that trigger is. And there's a million different ways, I would imagine. There's a little sheath that wraps around like a little rubber bag, and they just come like huge, gigantic enzymes and proteins. Now, they say all enzymes are proteins. Well, they're reconfigurations of different molecules to do different jobs. These are components that build things, proteins. They build. These are arranged in such a way that they break molecules apart. enzymes. They do catalysts, they're catalysts, and they chemically alter molecules instantly. And the product of that is given off as proteins, but also some other catalyst, some other bacteria intervenes and says, hey, now we got the stuff, this guy broke it all down for us, make me some muscle, make me some bone, make me whatever. And another bacteria creates the enzymes to package that stuff together, just like that. It's almost instantaneously how fast this stuff works. It's just stunning. You know, literally millions of years of chemistry can be done in less than 1 second when you have the correct enzyme. If you don't have the correct enzyme, you're done. And you just fall over dead within like 1 or 2 seconds, you're done. Enzymes do every single thing in your body. Every single chemical reaction in your body is facilitated by an enzyme. If you don't have the enzyme, that chemical reaction does not occur. And you are just nothing, and me are nothing but a bag of nucleophilic synthesis. We are continuously changing the molecules in our body, adding different minerals and so forth, and taking out the garbage. That's all nucleophilic invasion and substitution is all about. Now, in some cases, it comes in and it stays. That's in the rock case with my mud fossils. In life, it comes in, it goes out, comes in, it goes out, comes in, it goes out. That's just the nature of life. But once you die, or a creature dies in certain conditions that create mud fossils, and then this is the condition, and there are certain creatures that nobody can account for, they will stabilize, and the nucleophilic invasion happens, but and it substitutes in that thing. It doesn't keep doing it and doing it and doing it and doing it. It's done. Once it's done, it's done. It becomes stable with whatever chemistry it has to work with. And in this case, this became stable with, it looks like blood metals a little bit, you know, the irons. And then the other was just fluid-filled highway. All right, so let's just back through this again. We got a bacteria creating those little balls. The little balls turn into enzymes and proteins. This is what enzymes and proteins look like when they pop out of that ball. Every one of these little dots is a magnetic signature. And they all have to line up to whatever it's attacking, invading, breaking down, whatever it is. And every one of these is going to have a specific job. That's why they talk about, you know, breaking down starches and sugars and fats, how your body deals with salts. That's how it deals with all of those things. These have these are the chemists in your body. Well, this is the chemistry in your body. The bacteria is the chemist. They create those little polypeptide chains that go into that little ball, and they pop out like that only when they're needed. They just lay around. They don't eat, they don't poop, they don't do anything. So, and a million, hold on. Okay, be prepared to be shocked. In modern digital computers, all programs ultimately boil down to ones and zeros. It's called binary digits. It's either 1 or 0. And if you have enough of those, it means something. Well, guess what? DNA works with 20 different binary codes, not just ones and zeros. You see that? These are what we have for binary codes. There's 20 of them. These are all different little chemicals. And in a chain like this, they call it a polypeptide chain. And they may only be that long. They may be a zillion miles long. It depends on how much programming. But don't forget, we got 20 bits or 20 characters, and instead of binary, which is 2 ones and zeros, we got 20 to work with. So you can have 20. I would like to know from a programmer that knows what they're talking about, what factor of an increase in programming capability would there be if we had 20 characters to start with? Would there be any? Is it workable? I don't even know. I'm not a programmer. But when I see this, I do know about programming, just a smidge. I did technical stuff with equipment, so I know there's ones and zeros. And I know how digits move through the computer system with an A clock and a B clock. It used to be, I don't know what they do now, but it used to be there was a high and a low, and they only met at certain places when they were both high, and that's when data was transferred. Now, I don't know how they do it anymore, but it was a slow process. And I know things are going very, very fast now. But anyway, my point is, this is what's coming out, making into a ball, into a little tiny ribosome. And they're so tiny that you can fit millions inside of a single cell like this, an animal cell. These are the ribosomes, see? They're so small they don't even point out, they're just there everywhere. Most of them, though, collect on this rough endoplasmic reticulum right here, which is the entrance into the inner sanctum, the nucleus. So that's the protection device. These are all your ribosomes, which have all the chemistry you need. But you can have millions of these inside of one single cell, and still have all the mitochondria, the organelles, all the stuff that goes along with it. The Golgi apparatus, and some secretory vessels. There's certain things that secrete things. But all of that stuff is done by bacteria, basically. Now, do the bacteria live in these cells? I don't know. Do the bacteria create? I know the bacteria create the enzymes via ribosomes. And the ribosomes enter through these holes all over in the membrane. It just lets them in. The tiniest little thing you could ever imagine. A million fit in there. So they're just floating around everywhere. And if they're needed, something tickles it, almost like a key, a lock in a key, or a tickle on something that says, hey, I need you. Then they all just trigger, and they go to work. Unbelievably sophisticated system. Absolutely amazing. Now, I'm not certain where this is in the human body, or if even if it is a human, but I believe more than likely this is somewhere down in the lung area, where you got all these little tiny holes that can allow oxygen in and out so you can respirate. This, I believe, is the viruses that are attacking, and we are attacking back with our ribosomes that are going to pop open and kill these things. And I believe that's probably the fabric of the outside of the lung. I don't know. And it's probably from a washed out autopsy. I really don't know where this came from, to be perfectly honest with you, but this is what I see here, and I'm pretty sure that's the case. Let me show you something else, how cancer attacks. This would be a viral attack, in my mind. Okay, the way I see that, these are the attackers, these are the protectors, this is the tissue, probably a lung or respiratory area with all this webbing and allow oxygenation and so forth. So this is the point of contact. Now, I'm pretty sure this is what they claim cancer looks like, and it's a tumor that starts growing through our protective layer inside, and these tentacles take over, and it opens up a pathway for the tumor to work its way down through our vital flush. And apparently we're attacking back with some ribosomes to try to kill that thing. But it doesn't look like we're going to win in this particular case. Okay, this is talking about, this National Institute of Health is saying, although bacterial genomes, that's the sequence, have been traditionally viewed as being very compact, not a whole lot of sequences, relatively low amounts of repetitive and non-coding DNA, which means junk DNA. This view has dramatically changed in recent years. So now they realize junk DNA in there is not junk, and they realize junk DNA in the human body is not junk. It never was junk. And they're saying it recalls ancient attacks by viruses and stores them in our junk DNA. So apparently we can recall those sequences and build new systems against them or something. But the funny thing is, I agree that there are soil bacteria that are beneficial. And as a matter of fact, the probiotics I take have the soil bacteria built into them. Now, I know we went over this, but don't forget, viruses are specific quite often about exactly who they will attack and who they have no effect on whatsoever. So there's something in the chemistry of the tissues that are being attacked that allows that virus to take over. So there's either some bacteria living in there that it converts, or some cellular structure that does something to specific chemistry. Let's just go with that. And here's I said I took the homeostatic soil organisms. This is Garden of Life. Now, this is what we used over in Northern Ireland with Marguerite and her son, who had autism. He was just all twisted and distorted and had bang in his head. They were going to put him in a locked ward for the rest of his life. Now, they make a powdered one. Now, I take that stuff here. This is it right here. And it's not cheap. And you're supposed to take 3 a day. I take 2 a day. But I probably should take 3. But anyway, why homeostatic soil organisms? What does that mean? The HSOs, in primal defense, important probiotics you would get in abundance. You'd have them if your food came from soil untouched by pesticides, herbicides, and other widely used chemicals. Somehow they have removed all that stuff and are giving you 12 to 13 species, I guess. Now, so you're getting the stuff you would normally get from your foods if you weren't getting any pesticides, herbicides, or any of these other widely used chemicals. To replenish the value of probiotics, our pasteurized, irradiated, to replenish the valuable probiotics which our pasteurized, irradiated, and processed food no longer provides. There's a lot of food value that's just not there anymore. They're putting herbicides on them, which we eat, and that's herbicides in us. Now, primal defense formulas include 12 to 13 species of hardy, beneficial microbiomes, microorganisms in this probiotic blend with fermented organic grasses. And I'll tell you, I have no complaints, and I don't think anybody else does either that I've heard. Now, some people may not notice anything different, but let me just tell you what happened. With Marguerite, her son was going to be locked in a locked ward for the rest of his life. This was 6 years ago. I said, the only thing I can recommend is you get some good enzymes in them, and the only thing I know of that has the good enzymes that I would talk about is this stuff, because it saved the guy's life that I know about. Nice guy. And they would send him home to die. He started taking this stuff because a friend of his developed it or something happened. I don't know what happened there, but he knew about this, and that goes back 12 years ago. So, and he was good. He was healthy again in 2 weeks. Now, she started giving her son the primal defense, and she tried to do it in the pills. He wouldn't take them. They're very, very picky about what they eat, what they won't eat, and all kinds of things. So she ended up getting the powdered stuff and putting it in an ice cream cone, and he would eat it. And she was giving it to him, I guess, well, you have to do it regularly. There's no other way. It's got to be boom, 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 boom. Because this stuff goes bad. Well, it doesn't go bad. You use it up. Your body uses it up pretty quick. You're supposed to take 2 to 3 times a day is the recommended dose. Like I say, I take it twice. But she put it in the ice cream, and he started eating that stuff. And I guess it was less than a month he started talking. He'd never spoken a word. He started talking. He started being able to pick things up and hold things. He was never able to kick a ball or hold a cup of, and he was all distorted and screaming and humming and banging his head, biting, kicking, doing all kinds of things that he had no control. And within right now, he's virtually normal, and he's got girlfriends. He's playing football. He's doing everything that they all do. He's in and he's well admired by his peers now for coming back from where he was to where he is now. She says he's as they say over in Ireland, brilliant. And it's heartwarming to see that, because I'll tell you, this is a hard road to go down. If it's something as simple as this, and and all we need to do is get a bacterial database, and it'll show up one way or the other. I could be totally wrong, but I don't think so, because I just did a video with her, because she's been to The Hague and given a speech about this, and everybody's interested over there on the other side of the pond. And they're starting to do the research at the universities into regional areas with the different gut bacterial. They're going to start collecting data. So it takes a while. So it's going to happen, and once we know who's affected by what types of, because you could be affected by the water over in this area, and over here they're fine. It's as simple as that. If you got the wrong enzyme in the water where you are, the wrong bacteria, the wrong virus, or whatever it is, and it's not over here, well, guess what? These guys are vulnerable. Now, I work with fossils every day, and here's what they're talking about. These are retroviruses. These go way back. Genetic remnants of ancient viruses, infections that have become integrated into the genomes of their hosts. They are essentially fossil viruses. They were long ago, and now they are part of our DNA because we have evolved. Well, that's their take of this. This supports evolution, because then you see evolutionary aspects of retrovirus, and how this points to the fact that yes, there's no question we're evolving, and all of these things we evolved through are junk in the past in our DNA, and that's just not the case. In my opinion, it's not the case. All right, as I said, everything's about enzymes. The viruses, they can't synthesize proteins, generate energy, they can't do anything. They don't have ribosomes because they basically are ribosomes, only they take over cells and reprogram them. The other ribosomes go and create proteins or enzymes. These are programs. It's just a third type of ribosome as far as I'm concerned. All right, I'm just going to make my claim. They do a different job than bacteria and enzymes. These, in their own little capsule, are these viruses. But they need something to work off of. They don't poop, they don't eat, they just float around. And when they open up and attack something, they do just like an enzyme does. When an enzyme breaks food down and stuff like that, creates stuff to attack these kind of things, and also build stuff as proteins. These apparently are programmers. They go in and they reprogram the cells to spit out all kinds of these filaments. I think might be spitting out of here. They might be attacking in, I don't know, but there's certainly a lot of them. And they do overtake the mechanism of I believe it's of bacteria. And then they start creating their own. Now, where that first bacteria came from that created that virus enzyme, I'm going to call them virimes. They're virus enzymes. It's just exactly the same thing as an enzyme. They're viromes, virus ribosomes. They call it a virome now, is the virus of your microbiome. They call it a virome. And they're going to try to, well, I don't know if they're going to do it or not, but they should try to figure out what viruses populate a normal stomach or a normal body. And what we really want to find out too is what's in the sputum, in the lung stuff, because that's where you're going to most invasions apparently take place through the nasal, that type of thing, where you're inhaling. That's where you're going to get these things touching you, and that's where your membrane has to be good. And we talked about the membranes, we talked about everything, superbugs, yikes, run for your life. All right, we talked about the tight junctions. tight junction. Stay out of here. Don't come through here. But this whole thing has to float around and stretch and do all these kind of things. But it's layered with a layer of goo across the top, mucous membranes, basically. Anyway, I guess that's it for today, my friends. I love y'all. I think viruses are nothing more than ribosomes, but they are programmers and not builders and not breakers. They're programmers. That's the way I'm looking at it. Thank you. Bye.